Okay, thank you very much for coming, everyone. And thank you also to Jen and Anna for organizing these beautiful jornadas of the land. It's really amazing to see how the laboratory has evolved since, how long? 24 years or almost 25? Exactly. So it's a lot of time, and it's great that we have this every year organized, and it's great to be here. So today I'm going to show you some results of one of our main research lines that concern to our interest in understanding how plants move. So what are the ecological drivers of plant movement, and how we can understand better and parametricize models of plant dispersal that would be absolutely crucial and essential to know to predict, for instance, adaptive changes in a climate change scenario. And I don't know of you, but to me, the only plants that I know that can walk are the ants. And according to Gandalf, those are the only creatures that of the plant kingdom that can walk. And here you see the hobbits, and they are going to fight with Mordor and the orcs and the trolls. And really, real plants in the real world, they move, they stand still, but their genes move. And the genes are dispersed by many species, by animal pollinators or wind, or by the seeds by animal frugivores, vertebrate frugivores, or also the wind or rain or other dispersal agents. And we are quite interested in what are the consequences for the plants in different aspects of their life history of the contribution of animal interactors, animal mutualists, for performing those dispersal events. For instance, imagine that you are working in the field in a forest, and then you find a seed coming from a fleshy fruited species, like a seed of lentils that Elena was talking about before. And then there are two main questions that we need to be able to answer to try to understand quite well what are the biological and the natural history basis of plant dispersal. One is, which is the mother tree for that particular seed that reached that particular point in the forest, and who moved the seed over there. And those are two key questions that we have been exploring with different type of techniques, and there are many different techniques that you can use to quantify those dispersal kernel. A kernel is a mathematical function that simply establishes the probability of a given propagule reaching a particular distance. That would be a probability density function of the seeds dispersed from a given tree to reach a particular distance in the forest. And you can use telemetry, for instance, following the animal frugivores, and then using mechanistic models of the digestive process to establish which is the probability density function of seeds to be disseminated to a particular point in the forest. Or you can use traditional population genetic techniques to infer, like with FST or RST or GST models, to infer the connectivity among population. Or, for instance, some years ago, together with Godot, we introduced genetic techniques to try to assign a dispersed seed to the maternal tree where the frugivore consumed that particular seed. Or you can use inverse modeling techniques where you monitor the seed rain or the seed fall with seed collectors that Elena was talking about before, and then given the seed rain density that you get in the seed collector, if you know the location of the trees, of the plants, and their crop size, you may infer which is the most likely function, the most likely dispersal function that originated that particular dispersal pattern given the seed density resulting and the distribution of the tree sources. Okay? So those are the different techniques, and from many years ago we have been working in the Cazorla Natural Park with this beautiful wild cherry tree species, is the St. Lucis or Mahoma cherry, and we have been studying the dispersal of its seeds, also a lot of work with pollination. And then our setup have been setting those seed traps here in the landscape. This is a small population. The populations are quite fragmented in the forest, in the middle of the pine tree forest that grows in the rocky slopes of the area. So we have been monitoring the seed rain that we get in those collectors and then amplifying DNA from the seed endocarp that is diploid maternally derived tissue. We are able to compare these multilocus genotypes derived from macrosatellite genotyping with the distribution of those variants, the allelic variants that we catalog, for instance, amplifying the DNA from the leaves of the different individuals in the tree stand here. So we can determine that a particular seed captured in this particular point of the forest was consumed by the frugivore from that tree. So we get also those distribution of dispersal vectors. It's a dispersal distance vector, and also we capture a fraction of seeds that cannot be assigned to any particular tree growing in that particular populations, and we infer those trees are migrating, are coming from other populations. So we can establish those maps of the seed dispersal events, and then our next goal is to ask who contributed to those particular patterns of seed dispersal and highly heterogeneous seed rain across the complex landscape. So we identified the SCATs pertaining to 4 different functional group of frugivores: the small frugivores, the small passerines here, the large frugivorous mammals, including stone marten, red fox, genet, badger, and others, medium-sized birds like the thrush species, several thrush species consume the fruit, and also large-bodied frugivorous birds. So our idea is to link what is the seed rain pattern that we can sample in the heterogeneous landscape, and then assign the contribution to the seed dispersal kernel, that is the frequency distribution of seed dispersal events in terms of distance from the maternal tree where they originated. And I'm showing here in this inset those dispersal events originating from trees that grow in that particular stand. Okay, and then there we documented seed dispersal distances up to more than 1 kilometer in this large stand of trees. And then there is a fraction of immigrant seeds, around 20%, 18%, depends on the year of sampling, that we cannot assign to any tree growing in that population. And then we infer with maximum with Bayesian techniques, which is the source population of a set of potential source populations surrounding our focus population. So we know also the allelic richness of those populations, and then giving a propagule, a seed, whose genotype doesn't match any tree in that population, what is the likelihood that the tree was consumed, the seed was consumed and dispersed by the frugivore in that population with that particular genetic diversity, in that other one, and then we estimate dispersal events up to 17 kilometers from the original population. And you can see that those long distance dispersal events are really are contributed by a distinct subset of the animal frugivores, particularly the large-bodied species. And this is probably not a specific result of the prunus system, but is quite common in many animal dispersed plant species around the world. So any environmental driver that hits particularly strongly the large-bodied animals, like fragmentation of the forest or hunting, for instance, could collapse the seed dispersal kernel. And then a crucial aspect of those contributions is to assess what is the relative role of different species in contributing to those kernel. And here we distinguish just 4 functional groups, and we are interested also in assessing the roles of the individual species. But one conceptual challenge that we have when we study dispersal, not only plant dispersal but also animal dispersal, is what is a long distance dispersal event? How we can define that? And so far, the definition of the long-distance dispersal event, the long-distance dispersal concept are quite, let's say, fuzzy. I mean, they are study-specific. For instance, if we derive the dispersal kernel, we can say, okay, a long-distance dispersal event here would be any dispersal event taking the propagule beyond the 95th percentile of the distribution. But then we don't have, that would be very specific for the system, we don't have a comparative analysis. And then recently we developed a more conceptual definition, and it's based in that we need two reference framework to assess what is really a long-distance dispersal event. For instance, we have a population here that would be a tree stand here, an isolated population, maybe in a clear of the pine forest, and then there are different tree individuals, and we have here another populations, and a long-distance dispersal event can be defined with reference to two frameworks. One would be the geographic limit, the actual geographic limit of the population. The other be, for each individual tree, the actual limit of its genetic neighborhood size. So combining the geographic reference and the genetic reference, we can set up 4 different types of dispersal events. And only one of them would be long-distance dispersal event in the strict sense. That would be those long-distance dispersal events that take seeds both outside the geographic limits of the population and also outside the neighborhood, the genetic neighborhood limits, that for some species, like for instance neotropical figs, can extend way beyond, very long from the original population. So if we have those dispersal agents that combine taking the seeds outside the geographic population limits and also outside the genetic neighborhood of the population, then we'll be talking about a strict sense long-distance dispersal event. Other would be short-distance dispersal event within the same population, within the genetic neighborhood. We could have also some long-distance dispersal events that take the seed outside the geographic limits of the population, but still in some species with very extended genetic neighborhoods, still the seed can be dispersed within the genetic neighborhood. So that wouldn't be a strict sense dispersal events. So if we compute the size of the genetic neighborhood as a function of the variance in dispersal distances, in dispersal distance time, the generation time of the organism, we can assess the contribution, the relative contribution of dispersal events by the 4 functional group of frugivores within the population, within the genetic neighborhood, and outside and within or outside the geographic limit of the population or the genetic neighborhoods. And you see that in those plant species that are dispersed by a high diversity of animal frugivores, you see different contributions. You see some species that can be fundamental for the in situ regeneration of the plant with very strong contribution to both local movement of seeds, but also some species, some subset of functional group of species that are extremely important in maintaining in contributing the long-distance dispersal events that keep the cohesive metapopulation structure. We are now in our project that we are carrying now in Doñana. We are applying those techniques to understand the rapid area expansion of juniper, of Juniperus phoenicia trees that we are studying in the reserve. And for that we are using, and Elena already introduced the method, this DNA barcoding, where we amplify a 464 base pair region of the COI section, and then with that sequencing we compare to the barcode library for bird species, and we identify which is a frugivore species dispersing a particular seed. So in that way we are using the combination of DNA extracting from the endocarp to track the maternal tree, and then the animal originated DNA from the surface of scads or regurgitated seeds to identify who contributed to that particular dispersal distances. And we have done that with Juan P. Gonzalez Baro and also Juan Mi Arroyo and Carolina. And together with Alfredo Valido, and as a part of the PhD thesis of Nestor Perez Mendez, we have been using this type of approach to understand the consequences for seed dispersal of this narrow endemic Canary Island plant species, Neocamelea pulverulenta, which depends specifically on the large giant lizards in the Canary Islands, but then the lizards disperse a lot of different species in the Canary Island. And there the Neocamelea is distributed in the 3 central islands, specifically in Gran Canaria, in Tenerife, and then in La Gomera, where we studied the seed dispersal. And then we have an interesting scenario of defaunation. Around 3,000 years ago, when the Guanche aborigines arrived to the Canary Islands, they started to put a very strong predation pressure on the large giant lizards, and then they ate the lizards. And then 500 years ago, when the European Occidentals we arrived to the Canary Islands, we introduced the cats, rats, and also humans were consuming the large lizards. So we have this, comparing the Gran Canaria, the Tenerife, and La Gomera scenario, we have an increasing defaunation gradient because the very large giant lizard like Galotia goliath, and this is a cranium of a subfossil Galotia goliath, disappeared. So in Gran Canaria, we still have the large Galotia stellini. It's an endemic of the Gran Canaria Island, and is quite similarly sized to the subfossil form of stellini, the same species. But then in Tenerife and La Gomera, the large Goliath was extinct, and then in Tenerife we still have Galotia galoti consuming the fruits of Neocamelea, but performing a suboptimal dispersal because they are way smaller than Galotia stellini, and then Galotia caesaris is practically an insectivorous species. So we genotyped all the individual plants growing in 1 hectare plots. Those were many plants, and with the aid of Chris Dock, Cristina Rigueiro, we genotyped also the seeds obtained from scats of the lizard, and you can see here the dispersal kernels in Gran Canaria, where we have is the scenario closer to the pre-defaunation episode, and then advancing to the defaunated area. You see the probability density function of the dispersal event as a function of the distance from the plant, and you can see how quickly it collapsed in the most defaunated scenario. You lost all the fat tail of long distance dispersal event is completely truncated following the scenario. The lizards, the Neocamelea, is also secondarily dispersed. There are the lizards move we have been radio tracking the lizard, and they move over very small areas actually. But then the seeds can be secondarily dispersed by the common kestrels. How do the common kestrels disperse the Neocamelea seeds? The seeds are quite large. They carry the seeds here. You see here a kestrel taking a Galotia lizard, and they perch on the rocks, and the first thing that they do before decapitating the lizard, I don't know why, but they cut the head of the lizard like Predator. Have you seen Alien versus Predator? Predator also decapitates. They do exactly the same, but then before doing that, they eviscerate the lizard. So you can find seeds and seedlings of Neocamelea close related to the perching position of the kestrel, and this contributed dispersal events up to 2 kilometers beyond the maximum dispersal distance of the Galotia. So far we have been also together with Nestor and Chris Garcia and Alfredo working on the population connectivity graphs and to try to check what are the signatures of the faunation in terms of the cohesiveness of the population of this plant. Neocamellia is distributed in the basal, how do you say, el nivel basal, the basal level of the vegetation in the 3 islands, quite close to the coast. And we have been monitoring, we have been genotyping many plants from up to 85 populations. And you can see here the population genetic graphs. And then we are using that to try to understand how the defaunation is contributing to increasing the isolation and having a signal on the genetic structure. So if you use FCT, traditional FST analysis, FST compares the populations pair to pair, pairwise, and what we want to assess is the whole structure of the genetic covariance across the population. So for that, we start with a model where any pair of populations in any of the islands here can be connected with each other, and then we start pruning the links that link every 2 populations that would be directly related to the interchange of dispersal events among the 2 populations, and they are proportional to the conditional genetic distance between the populations that you are comparing. We start to prune out those links that don't preserve the genetic covariance. So finally, you get the structure of the network, of the genetic connectivity network that preserves the original genetic covariance that you have there. And then you can see that here you have a highly cohesive with small genetic distances among pairs of population in the non-defaunated island, an intermediate situation with distinct clusters of populations in the medium defaunated island with suboptimal seed dispersal by the medium-sized lizards, and then a much more disconnected scenario where you have a higher fraction of long links among the pairs of populations. If you have here a link, you may have 3 scenarios. The link is exactly, between 2 populations, is exactly the one expected from an isolation by distance model, an IBD model, or you may have a shorter link than expected under an IBD scenario, and that would be meaning that there is a frequent exchange of propagules among the 2 populations, or you may have a longer link than expected solely as a function of IBD, and that means that there is some process that is increasing the isolation between the 2 populations. So the fraction of longer links increases from the less defaunated scenario to the more defaunated scenario. So, I'm going in time. Okay. Why this is important? Because if we lose those interactions that support seed dispersal, either pollen dispersal or seed dispersal by animals, we can lose a very significant fraction of biodiversity, not only because of species extinction, but also because of the interactions that support those processes, they collapse. For instance, you have here the results of a model by Urban and Kitt, where you have different populations. This would be simply a map with the latitude and longitude and latitude, and you have here the locations of the different populations. They are connected by seed dispersal events. So depending of whether or not those dispersal services are preserved in those populations, you may completely lose all the connectivity in this meta-population scenario. For instance, if for some reason the seed dispersal services are collapsed in the population that I've marked here with a red circle, then you can have all this scenario completely disconnected. And this is nicely shown by the physics, working with statistical mechanics, where if you lose some crucial populations in the population network, you may get all the scenario completely disconnected. Here is the initial connectivity decreases very quickly as you increase the fraction of the nodes that are lost. And eventually, you get the total collapse simply well before losing all the populations. And if you lose some of the crucial populations, then you make that collapse even much earlier. So that's all. Thank you very much, especially to my colleagues working along this line, both Jose Antonio Godo and very especially Chris Garcia. They are for the work with Prunus, and also Nestor Perez Mendez and Alfredo Valido for the work with Galotia, and Juanpe González Barros. Where is Juanpe? Juanpe is here. And very especially to our technicians, Juanmi Arroyo and Chris Doss, Cristina Rigueiro over there. And thank you very much to all of you.